Hello, everyone. I'd like to thank the organizers for letting me talk here. I have a confession. I am not Dieter Egli. We had a last-minute substitution, so thank you for dealing with that. Um, my name is Michael Zuccaro. I'm a grad student in Dieter Egli's lab at Columbia University's medical campus uptown in Washington Heights. And I'm going to be talking today about a very special project, the human embryonic stem cells that we have. So why haploid human stem cells? What's so important about them? If you look at yeast, which we've heard a lot about today, and also recently there's been publications of multiple chromosomes in the yeast that have already been synthesized. And a lot of work is coming out really fast, and we really want to apply this to the human genome. But we come through a couple of problems. One, in yeast, they're haploid, and haploidy is extremely beneficial and attractive for doing functional genetics. Whereas in humans, you run into this issue of having diploid cells and heterozygosity and the like adds a realm of complexity to things. Also, the sheer magnitude of genetic material in humans compared to yeast. The haploid genome is just over 3 billion base pairs, whereas the yeast, I think, is only like 12 million. So if we want to write the human genome in an effective and cost-efficient way, I think regardless of the methods and the techniques we develop, if we can take the diploid material and cut that down to half, we can essentially have to do half the writing and half the workload, which I think would be very beneficial for something like the GP-Write project. So in 2016, our lab published a paper about the first derivation of human haploid embryonic stem cells. And these cells, you can see, are karyotypically normal and stable. These two images you show immunofluorescent staining of pluripotency markers, NANOG, SSEA4, TRA160. And this is important because you then have a cell type, you have a cell that you can use to interrogate the functionality of the genome in multiple different cell types from this one set of cells. And also we've shown that they are equivalent to their diploid counterparts, normal embryonic stem cells, in terms of differentiation. And here we have the 3 embryonic germ layers, the definitive endoderm, mesoderm, and ectoderm. And of special importance, I just want to emphasize, there were some talks before regarding ethics and the panels. Even though we refer to these as embryonic stem cells, there is no actual embryo that is created or destroyed to derive these cells. And I'll talk about that in a couple more slides. Just to elaborate, the differentiation, as you can see here, we do centromere staining. And so every chromosome will have 1 centromere. So a cell with 23 chromosomes centromeres is haploid, whereas 46 would be diploid. And you can see here that differentiating into each of these types, you do retain haploidy as well as functional diploids. So moving on to how we actually make these cells, we can do parthenogenetic cells, which means only the maternal genomic contribution is present. We can also do androgenetic now, which means only the male's genome is present. And so in both of these cases, you start with a human egg cell. And for the parthenogenetic cells, you activate them through an electrical pulse or calcium influx, and that triggers them to divide. With the androgenetic, you enucleate the egg, so there's no more maternal contribution. You then inject a sperm, and so now this only has the male genome, and that injection will also trigger the cell to divide. So now both in parallel, let's say, they'll proceed to develop in the same way a normal fertilized embryo would develop. So you have 2, 4, 8, 16 cell stage, and then it goes onto the morula, and then it becomes a blastocyst. And the blastocyst it has that iconic inner cell mass, that clump of cells there. I have a laser pointer. Inner cell mass, and that's where your stem cells are. That's what we isolate. And so we do this with these cells, and this is why we call them embryonic stem cells. But just again, to reiterate, because I feel this is very important, there's no actual fusion of the male and female genome. Each of these cells, there's no creation or destruction of a human embryo. Just that's a big takeaway from this. So we have these cells. They're pluripotent. They can differentiate. We want to know now, which we haven't seen before, are they compatible with gene editing, because that's something also for GP, right? If we want to be introducing these synthetic chromosomes into the organism, into the cells. So we did exome sequencing, and we found an interesting gene that we wanted to look at. And the mutation that our patient had, the donor for these lines, had a T instead of a C, and so we performed CRISPR. And you can see here, we successfully edited these cells to bring back to the wild type, and we also had an A mutated at the PAM site, so you know that homology-directed repair occurred and the repair template was successfully incorporated into the genome. And the other take-home message that I want to point out is that for us, when we do CRISPR, we typically have, with homology-directed repair, 1% at best efficiency. With these cells, we had almost 15%. This was highly efficient. We will be doing more experiments. This is just the first one. But I think this is also an extremely attractive feature for these haploid cells if they are, in fact, so easily genetically manipulated. And again, looking at them, they are karyotypically stable, and they retain their haploidy. One of the issues that we mentioned in the paper was that it's extremely difficult to first isolate the haploid cells. Right as soon as we do the derivation, there's a lot of diploid cells, and we do this through FACS sorting, which I'll talk about later on today. And we're hypothesizing, and again, this will be part of the pilot projects, that early on in the development stages, you're already getting diploidization spontaneously. And so we looked at the diploidization rate in our cells, and it looks to be about 1.5% per day, which doesn't seem like much, but if you don't constantly maintain these cells, you can easily wipe out your haploids within a month or so. And this is just an example of the sorting we do. And so your main peak in the middle, that's your G1 diploid cells. And as they go on in the cell cycle and duplicate their material, you get that peak to the right, which is your G2 diploids. Now, if you have haploids present, you have that left peak, which is your G1 haploids. Okay. And I'd like to stop there and conclude that the human haploid stem cells that we have can be a very powerful tool. You can functionally interrogate the genome in any type of cell, essentially. This is compatible with gene editing, and apparently it looks highly efficient. And again, this can essentially cut down the genome to half, which would, half the writing that we'd have to do, which would be great. And I'll be talking later about some pilot pitches, so tune in for that. I'd like to acknowledge our collaborators in Israel, Ido and Nissim, everyone at the CWRC who does the embryo work, Dieter, who put me on this and let me talk today, which was just unreal. Joao, who kind of exposed me to this through like secret experiments, and we got caught, and then Dieter was like, oh, you can do this, you can keep doing this. And then finally, the organizers and sponsors and everyone at GP Wright, thank you very much. And again, Dieter's not here, but we're highly interested in sharing these cells, and we think this can be a really great contribution. So here's our contact information, and with that, any questions, thank you very much.
So can you think of a selection that would reduce the 1.5% per day, was it?
Yeah, I'm going to address that. The pilot pitches that we have are stable haploid stem cells, and we do have a variety of experiments we want to perform and try to develop stable, because these are a great tool, we think, but we want to take it further and make it really a truly feasible option to use. Yes. Yes.
I'm a little mystified by the male haploid stem cells, because they wouldn't have an X chromosome, right? Or did I miss something? How does that work?
In males, we produce X and Y sperm, and so all the androgenetic lines that we do have are actually X. So the Ys, yes, that's correct. Yeah, yes.
That question, are you selecting out the inactive X when you do chemo on this, or is there a chance that you have inactive X? Because you have both active and inactive.
Right. We haven't looked into that yet, but that is something that's been brought up by our collaborators in Israel. Yes. So in the O site, they should be, yeah. Did you have a question? So we haven't. Well, we are in the process of looking at things like that. So with the FACS, the sorting, they're exposed to UV radiation, and we have to continuously do this. So what I'm doing now, and I think this was one of your questions last year for Dieter, Jet, was we're now in parallel doing these sorts with cells that aren't being sorted. We're going to be doing exome sequencing and the like to see if there's any changes and if there's a difference in the rate of mutagenesis and such. Okay, and with that, I have to end. Thanks again very much. Really appreciate it. Thank you.